So the struggle can be real when it comes to talking about osmolarity and tonicity and why they're different from each other, and so I thought it would be a good idea to maybe split this up into 3 different videos, and in this first one, we're going to talk about osmolarity. So before we get going on this epic saga, it will be really important for you to be well read on the prequels, so to speak. So if any of this stuff is still kind of a question for you, pause it real quick and figure that stuff out. None of them are that complicated, and you'll get them right away. But just for the sake of this video at least, I'm assuming that you're starting on the other side of this line. So molarity is just moles per liter, and we're comfortable with that. And osmolarity, as it turns out, is just osmoles per liter. And even though we don't really know what an osmole is yet, we're going to find out here soon. But it's probably easier to start off with comparing it to something that we know, like molarity. So let's say I have 2 identical beakers, and they're both filled with 1 liter of water, and I mix in 1 mole of glucose into both of them. And on the left-hand side, I want to talk about it and describe it in terms of molarity, but on the right-hand side, I want to describe the concentration in terms of osmolarity. Well, we know that the glucose is going to dissolve because water is polar and glucose is polar enough to dissolve with it, and they like each other. And if we were asked to predict what the concentration is going to be on the left-hand side, you would say, well, if there's 1 mole of glucose mixed into 1 liter of water, well, then it must be a 1 molar solution, and you'd be right. But what about on the right-hand side? It turns out that it's also a 1 osmolar solution. So far, it's not really different from what we've already learned, and it's not a big deal yet. But let's say that you decide to throw caution to the wind for a minute, and you get a little wild, and you throw in 1 mole of sodium chloride into each of these 1 liter beakers. Well, what's going to happen is you're going to see with your molecular goggles that the sodium and the chlorine got pulled off of each other, like forcibly removed from each other. You might be thinking, well, Logan, that's not impressive, who cares? Well, it was a really strong ionic bond that was holding them together up here, and now they're pulled off of each other. So what happened? Well, if we were to zoom in on either of these ions, we'll start with sodium. So if we zoom in on this sodium ion, if we remember that water is a very polar molecule and it has a negative dipole on one end and then very positive dipoles on the hydrogens, then it makes sense that all these oxygens are going to orient themselves towards the sodium because this sodium has a full positive charge on it. And it's almost kind of like a bunch of wolves surrounding an elk in one of those awesome National Geographic documentaries on Netflix. But over here, you're going to have the exact opposite happening. You're going to have all these positive dipoles of the hydrogens facing this full negative charge on the chlorine, and the oxygens are going to be as far away from that negative charge as possible. And all of these dipole-dipole moment associations that these oxygens are forming with sodium and that the hydrogens are forming with chlorine allow this very, very stable, very energetically favorable former ionic bond to be separated because it forms so many of these other associations that it becomes more energetically favorable for them to be apart. And what does that mean, though? And so now we know why they split up, but who cares? Well, let's talk about what that means for molarity and what that means for osmolarity. Alrighty then, let's make a prediction. So if I put in 1 mole of sodium chloride into 1 liter over here, and we're talking about this in terms of molarity, you would be very right to predict that it's going to be a 1 molar solution. And you are correct. That is the answer. Over here, and you probably see where I'm going with this by the way I'm setting this up, but if we put in 1 mole of sodium chloride into 1 liter, and then I ask you about osmolarity, it turns out it's going to be a 2 osmolar solution. So what gives? Well, remember when we put 1 mole of sodium chloride into our 1 liter beaker and it fell apart or dissociated? If you remember even farther back, the first time we added something, it was 1 mole of glucose. And I didn't say anything about glucose dissociating or falling apart. Not because I'm dumb or forgetful, although that should always be a concern with me. I didn't say anything about glucose falling apart because it doesn't fall apart. It does not dissociate. And when we look at water in comparison to glucose, we can see that glucose has the very similar polar covalent bond situations going on here. We have all of these OH groups all over glucose, and that's very similar to this OH situation in water, and where oxygen is a little more negative and hydrogen's a little more positive. And because of that, water likes to stick like the 2 opposite ends of Velcro to all of the opposite charges on glucose. And so these positive dipoles on water would like to stick to all of the
negative dipoles on glucose, and vice versa. All of the negative dipoles all over the water molecules would really like to stick to all of the positive dipoles all over glucose. But as it turns out, it does not dissociate. Glucose will not fall apart just because water is doing that. Water, sure, will come up here and it will attach and stick, but it does not pull anything off. This does not happen. It doesn't sequester anything. It does not come up to glucose and split it in half. It doesn't pull any of the hydroxyl groups off, which is very different from the way it behaved with sodium and chloride. Water completely sequestered sodium and chloride away from each other and turned what used to be AB, sodium and chloride, and turned it into A plus B, separate from each other in different situations. Glucose came in as one unit and stayed together as one unit. And therein lies the rub, my friends. Osmolarity and molarity are both very similar. They're both a way of describing concentrations, but you can think of molarity as if it's only concerned with how much went in, not about anything after the fact. So we have this much AB, that's all we care about. We have 1 mole of AB, 2 moles of AB, then we have a 1 molar or 2 molar solution if it went into 1 liter. But with osmolarity, it's not just about how much went in, it's about how much went in and what happened to it after the fact. And those are slightly different. So for example, what if I had 2 moles of AB and I put it into 1 liter of water? But we also know ahead of time we looked it up and we found out that AB, this molecule AB, will not dissociate at all when you put it into water, kind of like glucose didn't. Well, if I asked you about molarity, you would rightly tell me that if you put 2 moles into 2 liters, you're going to get a 2 molar solution. But if I put the same 2 molecules of same 2 moles, of AB, and I put it into 1 liter, and I ask you about osmolarity, keeping in mind that we already looked it up and found out that it does not dissociate, what would its osmolarity be? Pause it, figure it out on your own. It would be a 2 osmolar solution. But let's say that I've got a molecule called CD, and I look it up ahead of time and I find out that it does dissociate. Well, when I put CD into water, what kind of molarity am I going to have? If I'm asking you about molarity and I put 2 moles of it into 1 liter, you know I'm going to have a 2 molar solution. But if I put 2 moles of CD into 1 liter over here, and then I ask you about osmolarity, remembering that it does dissociate, what kind of osmolarity would it have? It would have a 4 osmolar solution. Now to wrap up this concept, let's get all of the situations on the screen at the same time and try and compare and contrast them. So if we remember that AB, the molecule AB does not dissociate in water, and the molecule CD does dissociate in water, then when we get all of these drawn out here, here's 1 of the moles, 2 of the moles of AB going in here, and if I'm talking about molarity, it's going to be a 2 molar solution, 2 moles per liter. And down here, look, CD, it did dissociate, but notice it's still a 2 molar solution, because with molarity, we don't care about what happens to them. We just know that we put in 2 moles of CD into 1 liter, that's a 2 molar solution. We don't care that they fell apart. Contrast that with osmolarity over here. We put in 2 moles of AB and we know they don't dissociate from each other, so here's 1 thing floating around, 2 things floating around. And so as far as things floating around are concerned, we have 2 moles of things floating around, so it's a 2 osmolar solution. But when you come down here, and we know that CD, the molecule, does dissociate, does fall off, water will sequester it and pull it apart, well now how many things do you have floating around in solution? You have 1, 2, 3, 4 things floating around in solution, so you have a 4 osmolar solution. So just to be clear, this whole time we've been talking about moles. That was 2 moles, 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 moles. And this isn't 1, 2, 3, 4 molecules we were putting in. It was 1, 2, 3, 4 moles. That's why it's 4 osmolar. The reason all of this is a big deal is because let's pretend instead of beakers that these were cells. They're all cells. Well, osmolarity is really useful to us because it describes exactly how much stuff is floating around inside of that cell. And there's something you've heard of before called, or you've probably heard of before, called osmosis. And osmosis is this really weird ability of water to move towards an area of higher concentration. So let's say this is my This pink stuff in the background is my water, and these little particles are my solutes. Well, if I have this thing running down the middle called a semi-permeable membrane, if that membrane only allows water to move back and forth, but it will not allow these little dots to move back and forth. So in other words, if I put a solute on this side, it stays on this side. Same thing over here. But water can go either direction. Well, if I do that and I separate the two different sides, and I've got a higher concentration right here and a lower concentration right here, if I look at it at time point 1 and then I come back a little bit later and check on it, my water will have moved over to the area of higher concentration. So after all of this, if you're still wondering why you should care, well, you care because we're not always dealing with beakers, and we're not always dealing with semi-permeable membranes inside of beakers. Sometimes those semi-permeable membranes are actually phospholipid bilayers, like in a cell. And there's extracellular fluid on the outside of cells, and there are intracellular fluid, or cytosol, on the inside of cells. And there are always things inside of cells that carry osmolarity inside of them, which I'm representing with these yellow dots. And sometimes those things can't cross the membrane. But water in most instances can kind of come back and forth as it pleases. And so if I have pure water on the outside with no osmolarity, and the only osmolarity is found within the cytosol, well, which way is water going to want to move? Osmosis says that it's going to come rushing inside of the cell, which would take the inside of take the cell's diameter and make it expand to get larger, which could pop the cell, break it, or it's called lysing the cell, which you don't want that to happen, right? Imagine if that happened to a brain cell. On the other end of things, though, what if we have way too much stuff on the outside of cells? It could be salts or protein or whatever, right? And if the osmolarity on the outside of the cell is very high compared to the inside of the cell, well, what's going to happen? Well, the water that's inside the cell or the cytosol is going to leave it in an abandoned ship and say, hey, the party's out here now. The osmolarity is way higher. And so the cell is going to shrink, or crenate is what it's called. So that covers osmolarity and why it's important. And in most instances, our cells are sitting in this kind of Goldilocks situation where the osmolarity is just right for them. In the next video, we're going to talk about tonicity and why it's different from osmolarity and what tonicity kind of means.